Hi, my name is Lily Mejia, and my presentation will be over the use of etoposide. Before I get started, I want to say one thing about topoisomerases. DNA topoisomerases are essential enzymes that regulate the topological state of the genetic material by introducing transient breaks in the DNA molecule. In the mechanism of action of etoposide is known to be a topoisomerase poison. Etoposide poisons the topo-2 cleavage complex and inhibits the second step to the reaction, which is DNA religation. This leads to causing DNA strands to break and induces an arrest in the G2 phase of the cell cycle. It is also known to happen in the S phase of the cell cycle as well. Topo2 can perform DNA relaxation as well as Topo1, catenation as well as decatenation, knotting and unknotting. These transformations are performed by passing on double-stranded DNA segments to one another. This will lead to irreversible double-strand breaks. By blocking topoisomerase 2, etoposide prevents it from relaxing DNA and causing the DNA strands to break. This prevents cells from dividing, which stops the growth of cancer cells. Topoisomerase 2 inhibitors, such as etoposide, stabilize the complex formed by topoisomerase 2 and the 5' cleaved ends of the DNA, thus forming stable, non-repairable, protein-linked DNA double-strand breaks. Cells are apparently able to recognize such DNA damage and, in turn, to eliminate the injured cells by apoptosis. It is known that high etoposide concentration can trigger the caspase-mediated apoptosis, which mainly occurs through the cytochrome C caspase 9 pathway. The side effect of this drug is that it can cause low white blood cell count, low platelet count, hair loss, loss of fertility, low blood pressure, nausea, and vomiting. Some facts on etoposide is that etoposide derives from podophylltoxin, a toxin found in the American mayapple. This drug is used in chemotherapy for treatments of ovarian cancer, lung cancer, testicular cancer, as well as various other cancers. In a research study, etoposide was able to kill 50% of the cancer cells in 72 hours, and the cell killing was preceded by the release of cytochrome C from the mitochondria. After reading a couple of articles, my question is, how is caspase playing a role in the etoposide mechanism of action? Before, I have stated how this drug played a role, its high concentrations played a role in the caspase 9 pathway, although I still do not understand the purpose of it. I know caspases are a type of protease, and they play a role in cell death, such as apoptosis and necrosis, although I'm still confused on how caspases and this medical drug are able to interact. My other question is, are there other uses of etoposide in the medical field besides cancer treatment?